S'engage on radiology quality management and quality control. About regulation and accreditation. In the United States, the federal government initiated radiologic and imaging sciences quality management regulations after the Radiation Control and Safety Act of 1968. The 1981 Consumer Patient Radiation Health and Safety Act added considerably to the scope of this effort, as did the 1990 Safe Medical Devices Act, the SMDA, and the 1992 Mammography Quality Standards Act, the MQSA. Today these acts and others relating to radiologic and imaging sciences are developed and enforced by the Center for Devices and Radiological Health, the CDRH, which is part of the Food and Drug Administration, the FDA, under the Department of Health and Human Services. The HHS in 1996, the Health Insurance Portability and Accountability Act, the HIPAA, commonly referred to as HIPAA, was enacted with the intention of simplifying standards by encouraging electronic transactions. This law also safeguards patient security and confidentiality, for which it is best known. Essentially, HIPAA established national standards for 4 categories. 1. Electronic record security. 2. Standardized electronic formats for record keeping. 3. Standardized electronic identifiers and codes for institutions, personnel, diagnoses, and treatments. 4. Requirements for confidentiality and privacy rules. Failure to comply with this law can result in significant legal penalties, up to a $250,000 fine and 10 years in prison. Of the acts currently in place, the Safe Medical Devices Act and Mammography Quality Standards Act have had the greatest impact on the practice of the radiologic and imaging sciences. SMDA mandates that any serious injury or death due to a medical device be reported. Mammography Quality Standards Act requires mammography facilities to be approved by the FDA. There are also voluntary accreditation procedures available, and they are used by the vast majority of hospitals in the United States. The largest hospital accreditation agency is the Joint Commission on the Accreditation of Healthcare Organizations, the JCAHO. Its approval is linked to many medical activities, most importantly to reimbursement by the federal and state governments and many insurance companies. Accreditation agencies establish quality standards, assess them, and provide certification that individual institutions have met the agency's standards. Quality management consists of a coherent system designed to monitor equipment performance through a variety of quality assurance and quality control standards or benchmarks. About quality assurance. Quality assurance consists of activities that provide adequate confidence that a radiology service will render consistently high-quality images and services. It is an evolutionary process that assesses everything that affects patient care. It may be medically, technically, or managerially oriented. Quality assurance includes evaluating activities such as interpretation of examinations, maintenance of equipment, performance of procedures, staff development, scheduling of examinations, and supply lines. The quality assurance process operates by identifying problems or potential problem areas, monitoring the problem, and then resolving it. Monitoring problems involves several steps, including establishing criteria, performing monitoring, and collecting, analyzing, and evaluating data. It is these steps of the monitoring process that are of greatest concern to the quality assurance radiographer. About quality control. Quality control is the aspect of quality assurance that monitors technical equipment to maintain quality standards. The concept of quality control is rooted in the need to stabilize the various equipment components of the radiographic imaging chain. From incoming line current through X-ray production to the processing of the radiograph, erratic equipment performance causes repeat radiographs and unnecessary patient exposure to radiation. The term quality control is sometimes used to describe the evaluation of individual radiographs according to acceptance limits standards. It is probably better to think of this image evaluation process as image critiquing or quality checking rather than quality control, although the quality control process is certainly a critical part of the critique evaluation. Technical expertise will ensure success in radiography when quality assurance is used to ascertain that the equipment is reliable. Because radiographic equipment changes as it ages, there are often great differences between the results obtained on one unit and those obtained on another. Table 31.1 lists the steps of the Focus PDCA method, which is an acronym for F. Find and define a problem. O. Organize a team to work on improvement. C. Clarify the problem with current knowledge. U. Understand the problem and its causes. S. Select a method to improve the process. P. Plan implementation. D. Do the implementation and measure change. C. Check the results. A. Act to continue improvements. Find, organize, clarify, understand, select, plan, do, check, act. Focus PDCA. The same imaging unit cannot be counted on to produce exactly the same beam for the radiographer to control and analyze unless it is properly checked on a regular basis. The system of checks to accomplish a measure of consistency in beam output is the quality control. Ensuring the quality of the radiographic image is a prime responsibility of the radiologic technologist. Every patient expects to receive the highest possible quality, or excellence, of service from a medical facility. No one wants to be exposed to ionizing radiation by a radiographer who is incapable of producing excellent images. It is important that quality control be seen as a method of controlling the radiographic image from start to finish. To do this requires a series of procedures. Unfortunately, some quality control begins with the last step instead of the first, with monitoring of equipment performance instead of an identification of imaging requirements. An effective program cannot be achieved simply by monitoring equipment performance, although this is unquestionably the most involved step. The thought process preceding equipment purchase is critical. Figure 31.1 describes a 7-steps total quality control system for unit procurement and operation. 1. Identification of imaging requirements. 2. Development of equipment specifications. 3. Selection of equipment. 4. Installation and acceptance. 5. Testing. 6. Continuing education. 7. Monitoring of equipment performance. This last step includes both the monitoring of processing systems and external beam evaluations. About purchasing equipment. Step 1. Identification of imaging requirements. It is not always apparent exactly what type of equipment is needed when new purchases are being considered. Historically, this decision was made by the chief radiologist or administrative technologist. Rarely are the actual operators of the equipment, the radiographers, consulted. The result has been that nearly every X-ray department owns a radiological monstrosity that is avoided by radiographers and radiologists alike. These units are often inconvenient, overly sophisticated, or unable to produce quality images. Careful identification of imaging requirements can prevent these problems. It is not necessary to be an expert in imaging physics and technology in order to be capable of contributing to the process of identifying the imaging requirements. Each member of the radiological team can contribute his or her expertise. The person who will make the final decision should interview the radiologists, administrators, supervisors, and staff technologists who will use the equipment. In many instances, the needs of the radiologist will determine the basic parameters for the purchase. Administrators must often impose financial and space restraints, although they can also create new funds and space when necessary. Supervisors are often able to provide important information on patient flow and staffing needs. After the radiologists, the radiographers are the most important persons to have input because their experiences with equipment provide a wealth of information. Step 2. Development of equipment specifications. Generic equipment specifications should be developed from the interview information. It is important that the person formulating the specifications has the technical background to state exactly what is needed to meet the imaging requirements. An imaging physicist should be involved at this point. The best specifications include detailed statements of what the equipment should be capable of doing. Here is an example specification. Maintain milliamperage linearity within plus or minus 10% to produce a consistent exposure when milliampere seconds is maintained but milliamperage is varied. End of statement. This must include a change from highest to lowest setting, a requirement that would be specified by a following statement. When the specifications are complete, they are sent to vendors for bidding. Step 3. Selection of equipment. The actual selection of equipment becomes simple if the investigation into the first 2 steps has been thorough. When the bids arrive, they can be compared for meeting specifications, cost, and service. The actual decision is then academic. A pitfall in the process can occur when radiologists in the institution show a preference to a particular vendor, thus weakening the bargaining position of the department administrator. In many institutions, this pitfall should be expanded to include interference by administrative officers unfamiliar with imaging equipment. Steps 4 and 5. Installation and acceptance testing of equipment. Installation and acceptance testing of equipment is the responsibility of the vendor and/or manufacturer. The quality control technologist must verify that the equipment specifications have been met. Normally this includes supervising the testing procedures and results. It is suggested that the exact methods for acceptance testing be included in the original specifications. The data from these tests will form the standard for all future quality control monitoring. Step 6. Continuing education. It is normally the responsibility of the vendor to familiarize the users of the equipment with its proper operation. With simple equipment, such as ancillary devices, the equipment manual may be sufficient for use in in-service training. More complex equipment should be demonstrated and explained by the vendor to at least 2 persons as part of the purchase contract. Continuing education must be an ongoing procedure. It is advisable to include both the quality control and in-service education coordinators in the initial training demonstrations. A good in-service program will include an orientation procedure as well as periodic updates on all complex equipment. Step 7. Monitoring equipment performance. The final procedure of a total program is commonly considered to be the whole of quality control. Monitoring equipment performance includes routine checks of all radiographic equipment. Although they are interdependent, monitoring can be divided into 2 parts. 1. Processing systems, and 2. External beam evaluation. 3 objectives for a performance monitoring system. 1. Monitor the quality of the image processing systems. 2. Measure the quality of the external radiation beams. 3. Specify faults within these systems to allow corrective measures to be taken. Evidence shows that a properly working quality control system will reduce equipment downtime and the number of repeated exposures. This will also reduce patient dose, patient waiting time, and resources, as well as increase confidence in diagnostic consultation and boost departmental morale. Responsibility for monitoring. Professional service personnel or medical physicists are not required to perform routine quality control procedures. Many procedures must be done daily, and to use these experts would be too costly. Radiographers, who are more available and knowledgeable about potential problems, should perform the equipment monitoring. An initial cost outlay for testing equipment is required. To maintain a program, staff technologists must be given time to perform the procedures and to evaluate them. About processor monitoring. Processor monitoring is utilized with film screen systems and is designed to permit an automatic film processor to fluctuate within set limits. The tests related to processor monitoring may be performed by anyone who has been oriented to the use of the equipment involved. However, only a radiographer or processor maintenance person should perform the corrective actions. About external beam evaluation. The second part of performance monitoring involves the evaluation of the external primary radiation beam. These tests must be performed by a radiographer who has knowledge of the physical operation of the equipment and all related accessories. Corrective action should be taken only by an authorized service person. Some monitoring equipment can be made very inexpensively, other equipment must be purchased. A dosimeter is an essential piece of equipment, and reasonable quality control cannot be performed without one. Computerized dosimeters are available that permit readouts and printouts of many quality control parameters from a single exposure. A room log should be kept to record test results, maintenance, and repairs performed on each X-ray tube and other system in each room. Diagnostic radiographic systems. The tests that should be performed on diagnostic radiographic systems should be done on a semi-annual schedule. The tests for X-ray tubes should include the following. Focal spot size estimation. An estimate of focal spot size is important so that it is kept within acceptable limits to ensure proper spatial resolution. There are 4 types of focal spot test tools. 1 line pair resolution tools. 2 star test patterns. 3 pinhole cameras. 4 slit camera. Both line pair resolution tools and star test patterns function by imaging a resolution pattern on an image. The image can then be analyzed to estimate the focal spot size. A pinhole camera permits measurement of the focal spot by creating an image of the effective focal spot on a film or on a digital image receptor, which makes it easier to process. While the pinhole camera provides a 2-dimensional image of the focal spot, the resulting images are difficult to obtain and interpret. The slit camera may be used instead to produce a unidimensional image of the focal spot that is perpendicular to the slit direction. This tool is recommended for nominal focal spot values less than 0.3. However, the use of the slit method is a difficult task to perform in the clinical setting. Half-value layer. The amount of total filtration in the primary beam is important for both radiation protection and image quality. Radiation protection is ensured by eliminating low-energy photons that are incapable of reaching the image receptor. Image quality is ensured by making appropriate contrast adjustments. Half-value layers are measured by using dosimetry equipment to detect the quality of aluminum filtration that will reduce the beam intensity to half the original value. Computerized quality control dosimeters provide a digital readout of the half-value layer. Half-value layer recommendations or requirements vary depending on the kVp. If insufficient filtration is present, the tube housing must be modified to supply additional aluminum filtration. Collimator, central ray, and bucky tray accuracy. The congruence of the projected centering light field and the central ray with the actual collimated primary X-ray beam and bucky tray is of critical importance for radiation protection. Radiation protection is ensured by avoiding repeated images due to overcollimation errors and by avoiding irradiation of tissue unless it will be imaged. A collimator test tool is simply a lead marker for each corner of the light beam plus crosshairs or a lead marker, placed several centimeters above the image receptor surface, for the central ray. A 2% surface-to-image distance error is allowed between the primary beam image and the light field size. This is a U.S. federal standard. The centering mark should be within 1% of the light field central ray. The positive beam limitation, PBL, mechanism should not permit the primary beam to be larger than the image receptor unless the override lock is activated. When these margins of error are exceeded, the collimator light or PBL mechanism must be adjusted. Distance and centering indicators accuracy. Many older X-ray units have inaccurate distance indicators located on tube support stands or overhead readouts. In addition, both distance and centering locks, stops, and detents may not be accurate. Assurance that distance and centering are accurate avoids the need for repeated expuries due to the inverse square law, misalignment of the central ray, and primary beam collimation. Distance indicators should be checked with a tape measure. Point 0 should be measured from the focal spot, which is approximately 1 centimeter from the bottom of the tube housing endca. Centering indicators can be checked by visual inspection of the collimator light beam, assuming a collimator and central ray accuracy test has been done. Distance indicators should be plus or minus 10%, and centering indicators should be plus or minus 2%. If the indicators are not within these limits, they should be adjusted. Ungulator or protractor accuracy. X-ray tables, bucky units, and tubes with ungulators or protractors must provide accurate readings for positioning baselines. They can be evaluated by using a large protractor for angle measurements in a level to verify that locks, stops, and detents are set to establish horizontal and perpendicular surfaces. Angles should be plus or minus 1%, with adjustments made when necessary. The tests for X-ray generators should include the following 4 procedures. 1. Kilovoltage accuracy. Kilovoltage settings tend to drift over time, primarily as a result of tube aging. Their accuracy is essential in maintaining image quality. The EI values cannot be predicted unless the kilovoltage peak is accurate. Verification and calibration of the kilovoltage peak ensures that technique exposure charts will produce diagnostic quality images. Computerized dosimeters provide digital readouts or printouts of average and single pulse kVp. If the settings drift beyond plus or minus 5 kVp of the labeled setting, the generator must be recalibrated. 2. Timer accuracy. Exposure time settings also tend to drift over time. Their accuracy is essential in maintaining image quality because exposure cannot be predicted unless the time is accurate. Verification and calibration of the timer ensure that technique exposure charts will produce diagnostic quality images. Computerized dosimeters provide digital readouts or printouts of the exposure time. Exposure time settings should be maintained within plus or minus 5% of the label. If the settings drift beyond this limit, the timing circuit or mechanism must be recalibrated. 3. MR over MAS, or millirentgens per milliampere second, and milliamperage linearity. Milliamperage settings also tend to drift over time, primarily as a result of tube aging. Their accuracy is essential to maintaining image quality because exposure cannot be predicted unless the milliamperage is accurate. Assurance is needed that technique exposure charts will accurately produce diagnostic quality images. Milliamperage cannot be measured by non-invasive quality control methods. Consequently, milliamperage station accuracy must be inferred by comparing milliröntgens per milliampere second measurements after both time and kilovoltage peak accuracy have been verified. Comparative measurements are made for the same milliampere seconds at different milliamperage and time settings to evaluate the linearity of the milliamperage stations. Computerized dosimeters provide digital readouts or printouts of milliröntgens per milliampere seconds. Milliamperage stations should be maintained within plus or minus 10% of each other, but some sources recommend maintaining the entire generator within plus or minus 10%. If the settings drift beyond the established limit, the generator must be recalibrated. For exposure reproducibility, generators must be capable of repeating exposures accurately. This is ensured by measuring the milliröntgens per milliampere seconds of several different exposures with the same technical factors. Computerized dosimeters provide digital readouts or printouts of milliröntgens per milliampere seconds. Reproducibility should be maintained within plus or minus 5%. If the readings are beyond this limit, the entire series of tube and generator tests should be analyzed to attempt to isolate the problem. More about digital radiography imaging systems. Digital radiography imaging systems include additional quality control tasks that are essential for their optimal operation. There is no standard quality control process that can be utilized by all systems, due to the variety of different digital imaging systems being used even within a single imaging department. Therefore, manufacturers' guidelines for specific QC procedures described in the operating manual should be followed. Many digital imaging system manufacturers and third-party companies offer hardware and software tools for performing quality assurance, which are intended to recognize equipment defects. Quality control tasks include rejected image analysis, which will be discussed shortly, exposure analysis, and artifact identification. About exposure analysis. Digital systems are more forgiving of exposure errors due to their wide dynamic range. Consequently, images can be produced with a greater range of receptor exposures, resulting in the need for exposure analysis, which should be conducted on a regular basis. Several metrics can be used to perform this task, including EI values, DICOM dose information, as well as dose estimation based on the technical factors used to acquire images. Data collected as part of an ongoing exposure analysis program can be compared with published reference levels for specific radiographic views, such as the National Council on Radiation Protection Report 174. Whenever collected data exceeds diagnostic reference levels, an investigation should be initiated. All findings and corrective actions should be documented and included in the rejected image analysis process. Organizing exposure analysis data in categories may provide additional information that is useful for quality control and quality improvement. For example, data could be organized by technologist, body part, and view, and radiographic room. About artifact identification. It is essential to identify digital image artifacts so that corrective action to eliminate them can be taken, including restricting equipment use. According to the American Association of Physicists in Medicine, the AAPM, the protocol for performing artifact checks involves acquiring an image using the calibration conditions recommended by the manufacturer and comparing it to a second image produced at 1/2 of the milliampere seconds used for the first image, with all other conditions identical to the first exposure. When images are analyzed, minimal post-processing should be used, with a specific window level and window width, as recommended per the AAPM protocol. The two images should be compared and evaluated for non-uniformities, such as grid lines, dead pixels, and dead lines. Dead pixels are non-functioning pixels that do not contribute to the formation of the image, and may even form dead lines when they comprise an entire row. Even though a small number of pixels may be non-functioning upon manufacturing, the radiographer is usually not aware of their presence. However, an increase in the number of dead pixels affects image quality. In addition to the artifact identification protocol described above, image receptor calibration and drop detectors artifacts require special attention. When image receptors are calibrated, images should be checked for artifacts because calibration files affect all future images acquired, until the next calibration is performed, and can even make any defects permanent until then. Additionally, it is important to verify that the image receptor is functional after a detector has been dropped, prior to using it for patient imaging. Many manufacturers install drop sensors, which require detector calibration after a sensor is triggered. Specialized radiographic systems, such as fluoroscopy, tomography, and automatic exposure controls, require additional tests. About fluoroscopic systems. The tests for fluoroscopic systems should include these 7 parameters. 1. Exposure rate. Fluoroscopic exposure rates are measured by a dosimeter exactly as diagnostic milliröntgens per milliampere second. The exposure rate should not exceed 5 Rmin. If the rate exceeds this limit, the automatic brightness control or other systems may require calibration or repair. 2. Field size accuracy and beam alignment. As with collimation accuracy in diagnostic radiography, fluoroscopic units should not be capable of irradiating tissue outside the image receptor area. In addition to the evaluation performed for a diagnostic unit, fluoroscopic units should display everything within 1 cm of the edges of the image intensifier tube, and the primary beam should be aligned to the center of the image intensifier flat panel detector assembly. Correction of the tube collimation, image intensification system flat panel detector assembly, or video display system may be required. Further testing by a service engineer is necessary. 3. Source to skin distance limits. Fluoroscopic units should not be capable of placing the tube target closer than 15 inches, 38 centimeters, or 12 inches, 30 centimeters for a mobile fluoroscopy unit, to the patient's skin surface. This is a U.S. federal requirement. Without making any exposure, the source to image receptor distance should be verified. If it is not within limits, the unit should not be used until a mechanical device is fitted to set a minimum source to skin distance. 4. Viewing system resolution. Because fluoroscopic resolution is much poorer than radiographic resolution, periodic assurance that it has not deteriorated is critical in maintaining confidence in diagnosis. A fluoroscopic mesh test tool or a resolution pattern test tool can be imaged to permit visualization of the maximum resolving ability of the system. If the resolution appears to be deteriorating, a service engineer should be consulted. 5. Viewing system contrast. Because fluoroscopic contrast is much higher than radiographic contrast, it is important to periodically determine approximately how low contrast a structure may be imaged. Special low-contrast fluoroscopic test tools are available that consist of 2 plates of aluminum with holes of 1 to 7 mm drilled in one of the plates. When the plates are sandwiched together and imaged on a fluoroscopy system, low-contrast evaluation can be made based on the smallest visible hole. 6. Image display systems. Fluoroscopic systems use video systems for image display because of the reduction in patient dose. Although a video signal generator is the test tool of choice, its expense and limited application in radiography usually prohibit its use for routine testing. A fluoroscopic mesh test tool or a resolution test tool and a piece of wire mesh are more commonly used. The resolution test tool can be imaged on the fluoroscopy unit to permit visual measurement of the system resolution. The wire mesh can be imaged to permit visualization of distortion. When degradation in resolution or distortion is observed, a service engineer should be consulted. 7. Automatic brightness control. Although there are a number of types of automatic brightness controls, the term is used here in a generic sense. These controls are designed to function like an AEC, an automatic exposure control, in that variations in subject density are automatically compensated for, resulting in a relatively uniform image brightness. A computerized dosimeter can be used to measure exposure after the beam passes through a phantom. When the phantom thickness is decreased by half, the exposure should be similarly reduced. If there is a great discrepancy between phantom thickness and exposure reduction, a service engineer should be consulted. About digital fluoroscopy units. The non-digital components of digital fluoroscopy units should be monitored using the conventional fluoroscopy methods described above. Digital components should be checked using a phantom that conforms to APM recommendations, and is capable of evaluating spatial resolution, linearity, image uniformity, dynamic range, as well as the variables of high and low contrast resolution. These tests should be performed on acceptance, every 6 months, as well as after a major repair. About tomographic systems. The tests for tomographic systems should include these 4 parameters. 1. Uniformity and completeness of motion. Tomography relies on the motion of the tube during exposure to produce a sectional image of the subject. Assurance of the uniformity and completeness of tube motion are basic tests. The test tool is simply a lead mask with a pinhole that is positioned several centimeters above the tomographic fulcrum. If the pinhole is centered to the image receptor and then imaged with a full tomographic motion, an effect illustrated by Figure 31.4, titled Tomographic Uniformity and Completeness of Motion Test with a Pinhole Trace Image is obtained. Both exposures depicted were erratic, as demonstrated by the uneven IR exposures along the entire pinhole image. Erratic tube motion is demonstrated by the uneven IR exposures, which fluctuated in intensity along a vertical axis with one or more darker nodes visible along it. If the motion were incomplete, there would not be a full length of the tracing. If the motion is erratic or incomplete, the mechanisms involved in the tomographic motion should be cleaned, rails, guide wheels, etc., and the test repeated. If the problem continues, a service engineer should be consulted. 2. Section depth indicator accuracy. Tomographic units are designed to provide an image that is sharp, possessing good detail, only at the fulcrum level. Assurance that the fulcrum indicator is accurate is essential in diagnosing the location of structures within the subject. Commercial test tools are available, or a penetrometer with lead-numbered steps can be used. When a tomographic image is made of the test tool, the number that was at the level of the fulcrum should appear sharp. If it does not, the fulcrum indicator should be adjusted to the appropriate level. Additional testing may be required to ascertain the proper adjustment. 3. Section thickness accuracy. The sharp section of a tomograph should be of a known thickness, depending on the total arc of the tomographic motion. Exact section thickness per arc is usually stated in the equipment manual. Commercial test tools are available, or an angled wire mesh can be used. When the angled resolution mesh is imaged, the section thickness can be determined by measuring the region of sharpness on the image. If the section thickness is inaccurate, the pinhole trace should be consulted. If it appears unacceptable, a service engineer should be contacted. 4. Resolution. Tomographic resolution is determined by imaging a resolution test pattern at the fulcrum level. The resulting image can be visually inspected to determine the resolving capability of the system. When resolution deteriorates, standard diagnostic quality control measures should be consulted first, then a service engineer should be contacted. About automatic exposure controls, AECs. The tests for automatic exposure controls should include the following 5 parameters. 1. Exposure reproducibility. The same reproducibility standards for diagnostic radiography systems are applied to AECs. If the readings are not within limits, the generator AEC circuits must be calibrated. 2. Ion chamber sensitivity. Most automatic exposure controls utilize 3 ion chambers and permit activation of various combinations during exposures. It is important to ensure that each ion chamber is equally sensitive by comparing the reproducibility of each chamber individually. Exact chamber locations should be verified by producing an image of the ion chambers by using the AEC without a subject at extremely low kVp. After this step is completed, the test is done by using a lead brick to block all chambers but the one to be tested. The AEC reproducibility test is then performed for the unblocked chamber. If all chambers do not respond within plus or minus 10%, a service engineer should be consulted to recalibrate the generator AEC circuitry. 3. IR exposure variation control accuracy. Nearly all AEC units provide controls to decrease and increase image receptor, or IR, exposure by changing the sensitivity of the ion chamber. These controls vary tremendously from one unit to another. Consultation of equipment manuals will provide the percentage of change each control is designed to produce. A computerized dosimeter can then be employed to obtain milliroentgens per milliampere second measurements to verify the intensity differences for each control. If the controls are not accurate, the generator AEC circuits must be calibrated. 4. Response capability. Each AEC has a minimum response capability which varies with manufacturer and model. If the AEC cannot respond at the minimum time, there is no assurance that diagnostic quality images can be produced. Response capability is best evaluated by using a computerized dosimeter readout or printout to determine exposure time for AEC exposures made with a small lucite phantom. As the phantom thickness is reduced, the AEC should produce exposures within plus or minus 10% milliroentgens per milliampere second of one another until a time below the minimum exposure time is used. If the minimum response time is greater than specified, the AEC should not be used until the problem is corrected. 5. Backup timer verification. Occasionally, Even the best radiographers will forget to activate the proper tube or AEC when this occurs. Patient radiation protection requires that AEC backup timers be functioning to terminate exposures and protect both patient and tube. When a lead plate is placed over the AEC ionization chamber and an exposure is made, the backup timer should terminate the exposure and a visual and an audible warning should occur. If the backup timer or signals fail, the AEC should not be used until the problem is corrected. Ancillary equipment. Ancillary equipment must also be monitored on a regular basis. About digital radiography image receptors. Digital radiography, or DR detectors. Most digital radiography vendors offer a quality control test tool for the periodic evaluation of their detector systems. The primary quality assurance parameter in these systems is known as detective quantum efficiency, or DQE. DQE measures the ratio of the transfer efficiency of the detector as the signal-to-noise ratio squared going into the system compared to that coming out. The formula is signal-noise ratio squared of the output divided by the signal-noise ration of the input. These values range from 0 to 1, with 1 representing a perfect system. Higher DQE values increase the ability to view small or low-contrast structures and are greatly valued in digital radiography systems. Computed radiography, or CR image receptors. Computed radiography PSP systems require regular erasure of the plates, as well as periodic inspection for cracks and surface markings. Erasure should be performed daily unless all PSPs are used every day. Visual inspection for cracks and surface markings should be performed semi-annually, with any plates creating artifacts removed from use immediately in the interval. Semi-annual performance of exposure tests is also recommended. About display monitors. Digital imaging display monitors require regular quality assurance to ascertain image quality and to monitor phosphor decay. These factors are sometimes referred to as monitor degradation. Both spatial resolution and brightness must be evaluated on a regular basis. Spatial resolution is evaluated through the display of a standardized grayscale tool. The X and Y display axes are evaluated by imaging spatial resolution test tools to evaluate point spread function, or PSF, line spread function, or LSF, and modulation transfer function, or MTF. This measures the matrix and pixel size accuracy. As with all quality assurance, the change in results over time is the critical test. Consequently, it is important to maintain a log of the test results for each monitor and to use it to predict when calibration and replacement should be scheduled. Brightness is evaluated through measurements of dynamic range with a luminance meter and unit of foot Lamberts, abbreviated small f big L, or candela, CD, per square meter. Dynamic range is measured by setting the greatest visible black and white level by using the monitor contrast and brightness controls. Readings from both levels are obtained with a luminance meter. Brightness uniformity is the difference between luminance measurements at the center of the display compared to the corner edges. The brightness uniformity specification is 15%. When a workstation has more than one monitor, the recommendation is that the luminance measurements match within 5%. About PACS systems. Because each imaging modality has developed quality assurance measures, the primary quality issue for a PACS system is the accuracy of the interface between the specific modality system and the electronic medical record, EMR, in the hospital information system, HIS, and radiology information system, RIS. Some experts in PACS management recommend that data from any system be held in a storage area, sometimes called the fix queue or penalty box, until the data can be matched with the modality, HIS, and RIS data or until human intervention resolves the mismatched data. This avoids problems with incomplete patient information being dumped into the PACs, where it adds to unfinished work lists, skews data, and contributes to misfiled patient information. It is also important that a quality check is done on the interface between each element of the PAC's system. This should include an item list that is developed with the manufacturer to assure the expectations of the clinical site are being met. One example that is often used is the proper setting for modality auto-send switches. These features are designed to permit real-time transmission of images from a modality to the PAC's system as the technologist completes them. However, service technicians and modality operators may turn them off to increase the speed of the modality functions and may forget to reactivate them, so physicians and radiologist assistants have immediate access to the images. About rejected image analysis. The careful analysis of rejected images has proven a valuable aid in diagnosing the radiographic process. Rejected radiographs are also referred to as repeat, retake, or double-dose studies. Even excellent rejected images studies need to take little time or effort to pinpoint conditions that may be easily corrected. Significant reductions in patient dose, radiographer time, and effort, and resources can be achieved with properly executed rejected image analysis. With the adoption of digital imaging systems, the physical evidence of rejected images is no longer present. Radiographers could simply delete images, or the rejected images would remain in the system until they were removed to free space, resulting in rejected images that were unaccounted for. Therefore, imaging departments realized that digital imaging systems require an innovative rejected image analysis. While early studies utilized manual collection and tagging of images from acquisition stations, more sophisticated programs have become available. These server-based programs are advantageous due to their ability to automatically collect, parse, and analyze data. Obtaining data. A rejected image analysis involves retaining all rejected radiographs, regardless of cause, for analysis. Any image outside the acceptance limits should be included. The most important rule when analyzing rejected images is to not discipline radiographers based on the information obtained. The analysis must be used in a constructive manner to improve working conditions and to point out areas where further technical training may be helpful. Radiographers must be informed of the intent of the study and asked to cooperate in a positive manner to improve both patient care and working conditions. Most radiographers are aware of numerous items they believe contribute to rejected radiographs and welcome an opportunity to find justification for their change. Incompetent radiographers should be disciplined through normal administrative procedures based on actual performance, not as a finding of a rejected image analysis. Failure by management to follow this suggestion may result in inaccurate rejected images data when images are not submitted for analysis. Analysis of data. The analysis of collected data begins with the classification and counting of images by cause of repeat, room, tube, and radiographer. Data should also be separated according to time, weekly being the most common. The compiled data should be examined for obvious problems. For example, a large number of images from a single room with an artifact in the same location might indicate a barium or iodine stain on a grid or table. Once obvious problems are addressed, the data should be examined for unequal distribution of repeats. The total rejected image rate is a valuable figure to monitor over long periods of time. It is calculated as a percentage of the total number of images produced during the period of the study. The total image figure can be extracted from institutional data in some cases or can be estimated by multiplying the number of examinations by the average number of images produced per examination. For example, a chest unit that produced 600 examinations in a week could be calculated to have produced 1,200 images, 600 posterior anterior and 600 lateral images. Rejected image rates may suffer from a startup effect where data may not be reliable for the first 3 weeks due to unfamiliarity with the requirements of the study or various psychological phenomena from stress to hypermotivation. Data indicate that radiographer rejected image rates should be between 3 and 10%, with the average at 8%. One study demonstrated the student rejected image rates average to be slightly above 9%. Data also appear to indicate that when the rate drops below 3%, there is a problem in data collection or an extremely wide acceptance limit in effect. It is important to realize that rejected image rates are affected not only by processing equipment, X-ray equipment, patient condition, and radiographer competence, but also by the acceptance limits of the radiologists. In some instances, a high rejected image rate may indicate extremely high quality standards in the institution, whereas the same rate may also indicate poor quality radiographers. Caution in interpretation is essential, and all aspects of the analysis should be carefully studied before conclusions are drawn. Record keeping. The total rejected image rates should be analyzed and documented monthly or at least quarterly. Documentation related to rejected image rates should be kept for the length of time required by applicable regulatory agencies. Additionally, corrective action in response to unusual rejected image rates should be documented, along with the observed results. About troubleshooting. Troubleshooting, locating problems when equipment malfunctions, is a diagnostic process. The same reasoning process that a physician uses to diagnose a disease can be applied to radiographic equipment. Beginning with the symptoms, the radiographer can work step by step through a problem-solving procedure to eliminate possible problems until the source of trouble is isolated. To summarize, Quality assurance consists of activities that provide adequate confidence that a radiology service will render consistently high-quality images and services. Quality assurance includes evaluating activities such as interpretation of examinations, maintenance of equipment, performance of procedures, staff development, scheduling of examinations, and supply lines. The quality assurance process operates by identifying problems or potential problem areas, monitoring the problem, and then resolving it. Quality control is the aspect of quality assurance that monitors technical equipment to maintain quality standards. The concept of quality control is rooted in the need to stabilize the various equipment components of the radiographic imaging chain. From incoming line current through X-ray production to image processing, erratic equipment performance causes repeat radiographs and unnecessary patient exposure to radiation. Equipment purchasing decisions should involve each member of the radiological team. Specific imaging requirements should be identified and equipment specifications should be developed. Once the equipment is selected, it is then installed and tested. Monitoring equipment performance is an integral part of quality assurance. It involves the processing systems, as well as the external radiation beams. Ancillary equipment should also be checked. External beam evaluations involve monitoring the diagnostic radiographic systems, digital imaging systems, fluoroscopic systems, tomographic systems, and automated exposure controls. Numerous tests on each of the systems are performed to ensure equipment accuracy. Ancillary equipment, such as digital image receptors and display monitors, are also evaluated. PACS system interface accuracy is also important. A careful analysis of reject radiographs is also a valuable aid in diagnosing the radiographic process. Significant reductions in patient dose, radiographer time and effort, and resources can be achieved with properly executed rejected image analysis.